proteins that catalyze chemical reactions by a molecule's substrate to interact with an enzyme's activity, therefore lowering the activation energy and the reaction becomes spontaneous. In this enzyme, our group tested the specific enzyme catalase, which can be found in chicken liver. We tested varying amounts of chicken liver with a substrate, hydrogen peroxide, to find at which rate this enzyme works best at. We hypothesized that if the amount of catalase is greater, then the reaction will be greater. But if the amount of catalase is smaller, then the rate of reaction would be smaller.
The materials used for the experiment were a laptop with Logger Pro, a temperature probe, graduated cylinders, safety glasses, a pestle and mortar, hydrogen peroxide solution, 3%, distilled water, test tubes in a test rack, chicken liver weigh boats, as well as an electric scale.
Cut the chicken liver using a razor blade. Put the chicken liver into a weighbow and find the mass. Grind the chicken liver using a mortar. Put the chicken liver into the test tube. Using the DPTP, add 1 milliliter. Using a DPTP, add 1 milliliter. Quickly insert the temperature probe into the test. Record onto Logger Pro until.
We will now be discussing the results section. This picture displays an example of what the reaction looked like as it was occurring. As shown in the picture, the catalase reacting with the hydrogen peroxide creates the bubbles of oxygen, causing the reaction to expand. This graph shows the relationship between the rate of reaction and time. The lines on the graph represent the temperature increase as the reaction between the catalase and the hydrogen peroxide progresses. The table on the right displays the rates of the catalase concentrations from the trials in order of increasing catalase mass.
In our lab, the change in rate of reaction throughout the trials was shown by the change in temperature. The higher the temperature, the more interaction is happening between the enzyme, in this case catalase, and substrate, in this case hydrogen peroxide. If this knowledge is applied to our results, then our hypothesis is disproven. This is seen in the multitude of rates dispersed throughout the data without any particular trend. However, although our hypothesis was disproven, there are a number of limitations that may have distorted the data. The first limitation was the inexact mass of liver. Although the liver was masked, afterwards it had to be grinded in the mortar. When the liver transferred from the mortar to the test tube, there was still residue left in the mortar, meaning that the mass of liver used was less than the mass of liver originally weighed and recorded. Another limitation was the inability to accurately record the temperature changes. The opening of the test tubes were simply too small for the temperature probe to be in the solution before the hydrogen peroxide was poured. The probe was only inserted into the solution after the hydrogen peroxide was poured, so the change in rate may have been affected because the time at which the probe started to measure the reaction may not have been consistent for every trial. Lastly, another limitation involved the unexpected scenario in which a controlled variable, the substrate hydrogen peroxide, became the limiting reactant instead of the catalase. Although our purpose was targeting the effective amount of catalase, because only a small quantity of hydrogen peroxide was used, it cannot be sure whether the rate of reaction was really determined by the catalase or the substrate.